I've already introduced myself. I'm Jay Olson, an investment research analyst at Oppenheimer in New York, and it's a pleasure to co-moderate this panel with Yury. Yury, I'll turn it over to you.
Yeah, thank you, Jay. And I would like to start with saying thank you to Sachs Associates for an opportunity to be here and talk about the topic which is very dear to my heart. As a quick introduction, my name is Yury Kukushkin. I'm one of the investment partners at Hevolution. I'm a scientist by my training, and I've been for more than a decade in venture capital in Boston area. And prior to that, I was for a few years in consulting. As a venture capitalist, I worked at Type 1 Diabetes Fund. It used to be JDRF T1D Fund, but now it's called Breakthrough T1D. And before that, I worked at 4Bio Capital and RBV Capital. Speaking about Hevolution, Hevolution is a global not-for-profit which is based out of Saudi Arabia and Boston. And we deployed and committed about $400 million in the last 2.5 years in grants and investments. And we are really trying to prolong the health span for the humanity. And I think it would be very helpful if all the panelists will introduce themselves. So I will turn it over to Ben Yaden.
Thanks, everybody. Can everybody hear me okay? Great. So my name is Ben Yaden. I am the Vice President and lead of the external innovation team that supports cardiometabolic health, but also women's health, aging, and aesthetics for Eli Lilly and Company. Background training is, again, I was a scientist in training for at least 13 years in discovery. We had a very strong musculoskeletal group for that time, for about 13 years. We were able to deliver some very unique programs and products to the clinic, things like obviously Forteo, Evista, but we had some really nice opportunities to explore additional mechanisms that would support mobility and general health and aging. At that time, I transitioned over into BD and Search and Evaluate, where I've been able to help sort of build out our new group that helps support broadly cardiometabolic health, but as you can imagine, now we're extending that into healthy aging and healthy living.
Hi, my name is Blythe Sather. I'm the head of research at a small biotech company called Therapeutics that's based in Durham, North Carolina and Seattle, Washington. Tune is actually an epigenetic editing company. We're a clinical stage biotech company. We actually have our first drug that's actually been reported in the clinic for hepatitis B using an epigenetic editor. So it's targeted to the viral genome to silence the virus. But I'm really excited to be on this panel today because we're actually also taking our epigenetic tools to use them in both cardiometabolic health and in type 1 diabetes. I think it's really important to understand, kind of particularly in light of a lot of the talk about the GLP-1 inhibitors, et cetera, kind of the epigenetic landscape that these drugs are inducing, particularly when we're thinking about it, longevity as well. I'm an immunologist by training. I've actually been in the cell therapy and the gene medicine field for about 30 years. So really very familiar with kind of the history of genetic medicines in oncology, and really excited about kind of this transition to epigenetic modalities. Thanks.
Hi, I'm Frederick Beddingfield. I'm the CEO of Rubedo Life Sciences. And at Rubedo, we are a private clinical stage biotech company. We've been in the clinic for about 1 year with a GPX4 modulator. It's in the first instance a topical formulation that's being applied to the skin, and we're studying a variety of conditions there, including medical dermatology as well as aesthetic conditions. We're developing a systemic form of the same drug, which would be used for cardiometabolic disease and neurologic disorders. And my background is I'm a dermatologist, and you might be saying, what the hell is a dermatologist doing at a cardiometabolic meeting? But the answer has actually been said already in the meeting. You've seen a relationship between cardiometabolic health and the skin. Also, the skin is just a great place to start. If you're interested in aging, in longevity, you can see the skin, you can access the skin, you can assess the skin. People will let you do biopsies a lot easier of their skin than the liver. So we started in the skin. It's not our final destination, but it's a great place to start. It's a good business. And I think it also, given if you have positive results there, kind of validates the approach you're taking to your systemic drug as well. So I'm also on the board of another company, Neurodon, which is a cardiometabolic company focused on SERCA activator for obesity, diabetes, and sarcopenia.
Hello, nice to meet everyone. My name is Robb, and I'm the investment principal at a single LP-backed fund called Lifespan Vision Ventures. Our thesis, similarly to Hevolution, is to invest in therapeutic approaches that modulate the biology of aging as a way to enhance functional health and resilience across that aged population. I think what we have recognized is as people are getting older, they are comorbid, and so we're interested in the biology that sits within that comorbid patient setting, and perhaps that's more directly relevant to aging. We can invest globally from pre-seed through to Series A, and Cardiometabolic is a strong proportion of our portfolio, given I think there's a bit of a clearer line of sight to that sort of comorbid patient setting, but we also do immunology, oncology, and neuro. Previously to setting up the fund, I worked in the advisory side, specializing in longevity. Again, so I've spent my entire professional career, and I've seen the progression of the field starting to become more mature. Great to be here. Thank you.
Thanks, Harry. Jyothis George. I had the privilege to lead SGLT2 clinical development with Lilly Alliance. When I was speaking to your previous colleague just earlier today on the great alliance we had, and led the expansion of that into heart failure and kidney disease, to Harry, to your point about cardiometabolic driving health span. Then I had the opportunity to lead the GLP-1 programs, including the very first cardiovascular outcome trial for GLP-1s before it became a thing. Currently, I lead NodThera as its Chief Medical Officer. For the last year, we've focused quite sharply on cardiometabolism, particularly ASCVD and inflammation. And just last week, we published our data in the Journal of American College of Cardiology, and looking forward to getting our phase 3 underway in cardiovascular in the next year. What am I excited about? I mean, just like the early days of SGLT2 and GLP-1, this field of lifespan, health span is at the very beginning of a journey. What does that mean? We don't have a clear path to patients. We do not know what endpoints we should register for. We do not know who will pay for it. We do not know why anybody should prescribe it. We do not know why patients should take the drug. So a lot of things to figure out, and I think it's a great opportunity for the community to come together and figure it out and then solve some of the world's biggest problems.
Hi, I'm Paul Rubin. By training, I'm a physician, internal medicine, clinical pharmacology. I've been in large and small pharma and biotech for more than 35 years. Been involved literally with dozens of drugs that ultimately were approved. Interestingly, about 6 years ago, I was very seriously considering retiring. And I was introduced to Eric Morgan and Kristen Fortney, who were the founders of BioAge. And they told me about their platform, which essentially is they had identified blood samples up to 50 years ago, databases where they had ensuing healthcare records from that 50-year period, many times up until these patients' deaths. They were able to take these samples and run modern-day proteomics, metabolomics, genomics, and actually determine signatures that were actionable, that correlated with all the diseases that these people got as a function of age. As a result, they were able to discover very unique targets. One of those targets is NLRP3, which is one that we're working on. Just the platform itself got me very interested, and it occurred to me that probably the number one risk factor for most diseases is aging. And as I got older, this became very directly relevant. So I started working with them and ended up now being there for 6 years. Looking at it. So I believe that what we're doing is becoming actionable, although as other people have said, I mean, when you're trying to develop diseases strictly for aging, there is no clear pathway. So as a result of that, we have to determine a direction that is actionable, that's something that you can develop.
And it's probably more related to the diseases associated with aging initially and then eventually going to aging. Or else, as mentioned before, there is no practical way to go forward at this stage. So, nice to be here.
Excellent. So maybe we can start with a kind of an elephant in the room type of observation. GLP obviously changed the way we think about metabolic health, and there is a lot of health span data coming out of large clinical trials with GLP. So in that regard, I'm curious, how exactly how should we think about investing in other types of health span opportunities? What lessons do we learn from type 2 diabetes and obesity kind of playbook and GLPs, which can be applied in other areas? And kind of what is next? And maybe I will turn it over to Paul. And Paul, if you can speak actually a little bit closer to the microphone, I think it would be also super helpful.
Sorry, it's difficult for me to hear. Were you addressing that to me? Is that,
Yep.
Yeah, great. So, I mean, when you look at the effect that GLP-1s have had on the world, it's phenomenal. And it's taught us what a drug that can actually have an effect on weight loss can actually do for the world, literally. But it's also taught us the issues associated with administering GLP-1s, where it's clear that if you can take them, you could tolerate them, and you could stay on them, not only is your health, but your life is remarkably changed. But what are the lessons that we've learned from that? One, does everyone want to stick themselves with a needle? That's one thing, which may or may not be the case, and there's probably a sizable percentage of the population that aren't interested in self-injection. The side effects, in many cases, make it intolerable for certain folks. And then where fat loss is clearly achievable, and you can do it, there are other things that occur as a result of that weight loss, which includes loss of muscle mass, loss of bone density. So I think when we're thinking about things like weight loss and certainly treating metabolic disease, we have to consider not only the positives and the negatives and try to develop things that will compensate for the adverse events of this. Although obviously on balance, the positives are greater than the negatives. If we can also overcome the negatives, then we're going to have an even broader use or broader utility of these types of drugs.
Thank you, Paul. And maybe I will turn it over to Ben. And I do remember, Ben, you just mentioned, bone health and, also muscle health. And I'm curious if, you want to comment on this or anything else, feel free to.
No, absolutely. It's interesting, I guess, part of my career is really sort of a bit bipolar, right? I spent the first half of my career trying to actually increase body mass. And then I think now, so the second half I've been trying to get it off. So I think, there's a balance to how to really understand this. And I think what's great about the GLP, at least from pharmacology side, from just the basic mechanism, it's got us talking about longevity again, right? It's not a perfect drug by any means, but I think what we're seeing in terms of benefits on sort of a daily life span in terms of sleeping, osteoarthritis, obviously chronic kidney disease and heart failure, and it's opening those questions up to longevity. Now, we absolutely know there are potentially deleterious effects, or at least effects that are unwanted in the musculoskeletal system. So what's great is it's opened a door for us to revisit those mechanisms. So as we want to think about, improving skeletal muscle quality, improving bone health, I think it'll help us truly understand putting this puzzle together, or this jigsaw puzzle together, of what a polypharmacological approach could look like to eventually treating, again, aging and longevity for different types of patients. Again, GLP is going to be great for certain sets of patients, but I think as you get into more frail individuals or different sort of distributions of fat, I think there's going to be opportunities there as well. But I think it's nice is it's got the conversation back to what I originally worked on, which was, again, improving things like bone. And I think hopefully we don't just focus on, whether it's the architectural support or just the functional support of muscle. I think one example that we have with bimagrumab is the case we built around it was bimagrumab was not only providing increased muscle mass, but it was also providing metabolic support, right? So we're pulling other functions in to say, hey, yeah, it's really tough to understand function. It's really important to understand activities of daily living, but let's not ignore these other benefits that skeletal muscle can provide. So again, I'm a big fan, obviously, the incretins piece. Again, I know it's always a polarizing topic in this space, but I think it's a great way for us. It's a gateway for us to start having these conversations, and it's providing us the resources now to start thinking broadly. And so with our clinical trials, with everything that we're doing now, obviously we're enriching those with different types of patients, maybe patients that are a bit older, maybe patients that are perimenopausal, postmenopausal, loading them down with biomarkers and other exploratory endpoints to truly understand how we can segment and triage those patients out to eventually treat them better. And I think somebody mentioned earlier precision medicine. And I think every time we do an obesity trial, one, we're learning a lot, but two, we're also learning that, hey, this could be the population that could use a bone drug, or could use a bone plus muscle drug, or maybe a joint drug, or something on inflammation. We've been doing on that. So, sorry, I've had a lot of coffee, so I can ramble on forever, but I'll let my other colleagues speak to it as well.
I want to pick up on something that I think, Harry, you brought this up in our prep discussion, and it's really kind of the idea of a foundation, or I think you called it a blueprint. Obesity kind of sets the stage for aging in terms of defining specific indications that can be pursued for clinical development and regulatory approval, and eventually reimbursement, and the consumer-driven nature of demand and access for obesity drugs, where patients are actually potentially willing to pay out of pocket, which also could apply to drugs for longevity. So maybe if I could ask you to elaborate there.
Yeah, of course. I'd say, I think Ben just mentioned it briefly, but the idea of this polypharmacology, always we'd consider it to be beneficial pleiotropy. And I think prior to the GLP-1, the pleiotropy term was looked pretty disfavorably. And I think that this has kind of set a precedent that having something with a beneficially pleiotropic effect or a polypharmacology effect actually is, the market will respond well to that if there's that functional outcome tied with it. So I think that's been a bit of a change, because a lot of aging-related mechanisms or targets or drugs have always had this idea of being beneficially pleiotropic, and now that has been validated as something that's seen up unfavored by the market. I think also what's interesting is the idea of doing obesity-associated indications to build upon sort of a backbone of GLP-1, and I think that also sets a precedent for the idea of multifunctional therapeutics. And this is something that we spend a lot of time thinking about, is in complex disease, of which most chronic diseases are, you're probably going to have to tweak more than one node at the same time in case there's a redundancy in the biology. And I think having sort of the GLP backbone with GLP plus something else in a combo or a bispecific, I think sets further precedent of that. And then lastly, just clearly this has kind of opened the door for a direct-to-consumer perspective, and clearly there's a consumer demand going back to antiquity around anti-aging, or whatever that may be, and I think that's something that the field can probably lean into, but how that occurs over time, I'm not sure, but I think it's an interesting thread to follow.
Great, thank you, Harry. Jyothis, anything you'd add?
Yeah, one of the things that we haven't touched upon is the importance of preserving brain function. So it's not just how many years do we live, it's actually it's not just adding days to life, it's adding life to days as well, right? With that thing, there is a lot of excitement, investment, vaporware on lifespan and longevity. Yet my hypothesis is that we actually have to have the right drug for the right patient at the right time. And one of them actually is making sure that there is actually very good CNS penetrant efficacy that you can show early. And we are all talking in the context of GLP-1, but we haven't talked about how semaglutide, wonderful as it is, it failed to show improvement in cognition. So hence, I think Ben mentioned inflammation, and Paul and I both work on NLRP3 molecules. So specifically, our company's been at it for 10 years, and we've actually shown that by taking patients with Parkinson's, you can show neuroinflammation going down. Yet the sequence of indications might be different, that you actually have to get maybe into cardiovascular first, because these benefits on longevity and cognition take large number of patients and a long time to come. So I think the thing therefore is not just what is good biological plausibility, it's having good drugs that go with it, the right drug for the right compartment you want, but also indication selection sequencing, so investors like Yury and Harry can have a reasonable return in a reasonable time point as well.
Great, thank you, Jyothis. Blythe, would you like to comment?
Yeah, I mean, I think it's really exciting when you look at a lot of these drugs, particularly GLP-1. They didn't really set out to be longevity drugs, but we're really finding that the epigenetic changes that they're inducing from the clocks to the kind of inflammasome and then of kappa B to adipose chromatin remodeling, all of these things, we really need to understand, are they correlative or are they causative? Because if they're causative, you can use the drugs such as the drugs that Tune is developing, the very precise epigenetic changes that we can induce to actually get right at the heart of what's causing the positive benefits of these drugs and get around the negative benefits of these drugs. So, really when you're starting to gather, around these kind of drugs, any kind of data, looking at the epigenetic landscape in these patients and really trying to understand over time as they take their drugs what those changes are, can give us kind of new places to target and new ways to kind of get at this longevity problem.
Excellent. Thank you, Blythe. And I think that's a perfect segue into our next question. So let's explore a little bit the underlying biology of aging and how potentially it's kind of interesting from the development perspective. So if you think about the way how we age, right, there are different frameworks which people use. I mean, the most common one is hallmarks of aging and no hallmarks of aging, such as, inflammation, epigenetic drift, senescent cells, mitochondria function, proteostasis, and so on and so forth. But I mean, I think we shouldn't be like too reductionist in that regard, especially we discussed already GLP, right? Because GLP on its own doesn't affect directly any of those particular hallmarks of aging. It's not, working directly on whatever mitochondrial protein quality control. But at the same time, it actually touches many of them as a result. So in that regard, like everything which goes together with aging biology kind of fits to this question. So making a step back and looking at those mechanisms in terms of how we age, what in your opinion, where do you think we are ready already to explore potentially therapeutically those areas because there is enough evidence, and where do you feel that we actually are not ready yet because it's more a scientific experiment and more validation is needed? And maybe Paul, I could start with you as the first speaker.
Yeah, well, let's start with the first thing that you mentioned, which I think is unique in the fact that there is data not only supporting but confirming the fact that it is relevant for diseases associated with aging, and that's the concept of this inflammaging. As people grow older, their inflammatory system tends to change. You become less lymphocyte, more neutrophil predominant. And when you look at multiple diseases, there's clear correlations with the innate immune system being turned on. And it not only is it correlative, but now there's also data suggesting the third part of Koch's postulates that if you remove it, people do better. A good example is, for example, with the antibodies to interleukin-1 beta that have shown that your cardiac outcomes are better if you can block them. So the concept of trying to manage this inflammaging process or this turning on of the innate immune system is something that is actionable as we speak right now. There are multiple approaches to it, things that are ongoing in the clinic. There's data supporting the fact that if you do suppress inflammation, you'll have a benefit in terms of multiple diseases. And when you look at the number of diseases that correlate with an increase in inflammation, it's many of them. We talked about neurocognitive dysfunction, cardiovascular dysfunction, joint health, many different things that seem to be not only correlated, but it seems to be causative. So by approaching inflammation as one, I think there could be huge benefit related to this.
Yeah, thank you. This is super helpful. And Jyothis, maybe I will turn it over to you. And obviously you also worked a lot on kind of similar types of mechanisms. How do you think about what's right and what's not?
Yeah, at least for me, it's you actually ranked a number of possible pathways. And for me, it's in the road to hell is paved with biological plausibility. And that's a pitfall that we can all fall into. But I think, violently agreeing with Paul that I think, inflammation is a different order of magnitude in terms of evidence, because we have 30-year follow-up from the Physician's Health Study, for example, on cardiovascular outcomes. And much of this day we've spent talking about BMI and weight. If you go from a high BMI to low BMI, versus you go from a CRP more than 2 to CRP less than 2, your CV risk reduction is almost identical, and you have no nausea and nothing of that sort to deal with either. So, and therefore, this is not no longer a good biological possibility. There is good clinical conviction. In addition, I think what we have to actually bear in mind is that it also matters how you actually modify these risk markers. We've actually learned, again, with multiple pathways modulating CV risk and neurological risk, that it's not just the end, but it's also how you do it. And that's where I think if you actually block the complete cytokine pathway with inflammation, injectable IL-1, IL-6 will do the trick. Like Paul said, you will get better CV outcomes. But what the sweet spot would be if you can actually modulate bad inflammation while preserving the ability to fight infections. And that's where agents like NLRP3 come in. And we've already seen that patients who are treated with our drug, for example, can mount a normal infection response. Otherwise, you will not have a long life. You will actually suffer with infections, right?
Yeah, excellent. So maybe, Hari, if you can also share how you think about, this topic, and if you have any kind of frameworks which you utilize to think about the biology of aging.
Yeah, of course. I think I'd sort of preface it with saying that I don't believe there is a singular silver bullet for what would define a longevity biotech company. I don't think there's one data point that will inherently give that a tick. So it's a constellation of different things. I think, the paradigm of the hallmarks of aging, I think, is a useful place to start as a way to understand the field, but I don't think it's the be-all and end-all of the field. As you say, it's a slightly reductionist approach to aging biology, and the hallmarks do increase year on year, it seems. That being said, I think, has been referenced already, I think different hallmarks, they're not created equal in sort of their effects. So I think, in the way of inflammation, and then also in within the epigenome, I think they probably have a different effect size from hallmark to hallmark. But the way we look at it is, we probably, when it comes down to it, it's like, okay, what does your drug actually target? And then what are the effects of that, of the modulation of that target or targets on a suite of hallmarks of aging? And so we're looking at approaches that we think can modulate more than one. And then going beyond that, do you actually have the evidence to suggest from a translatability perspective that you can restore biological function in an age system? And can you improve resilience in whatever way you seek to determine that? So there's an element of the fundamental biology being relevant for aging, but then can you also substantiate that in an age system as well?
Fred, can you share your perspective?
Yeah, I think a lot of good points have been made here about the hallmarks of aging, and not to be overemphasized that no singular one is going to be the pathway to biological health and longevity for sure. At Rubedo, what we've been doing is really taking modern techniques to analyze cellular data, single-cell RNA sequencing, multi-omic data, and say, what's the difference between healthy cells and non-healthy cells? What's the difference between aged cells and young cells? And trying to look at those differences and see how can we impact that. And through that, we found GPX4 to be a pathway that when one can modulate that pathway, you can either trigger a senescent cell to undergo a type of apoptosis, or if a cell is not quite all the way to senescence, you can trigger it to sort of get healthier, regenerate, rejuvenate itself through sort of a stress response. And I think that's interesting. We're seeing now, there's the theoretical, how are we going to treat aging and age-related diseases, and there's a practical. We're in the clinic. We're seeing that when we use this drug on the skin, we've treated psoriasis, atopic dermatitis, had a dramatic reduction in actinic keratoses, and we're improving aging skin. And that's important. And we're seeing epigenetic changes. We're seeing changes in practical things like collagen. So there's a lot of pathways here. I think it's great that everyone is really looking and casting a broad net, because sometimes you never know what the final impact is going to be. But at the end of the day, you've got to have clinical data. And I'm more of a clinician, I'm a dermatologist, I'm a drug developer. So I focus on what are things that we can do. And I love what the GLP-1s have done because, they started with an indication and then they just expanded it. And they are the best case for a longevity drug today. And kudos to you all. I think you have changed the world. And that's, I think, what we all up here are hoping to do. And we've just got to keep exploring these various pathways and seeing what we find.
Yeah, absolutely, Fred. I mean, that's I think the reason why I really felt strongly that we needed a little bit to discuss underlying biology, because it kind of lends itself to actually this exploration from disorder to disorder. But we will talk about this super soon. Let us finish a little bit with the biology. And Blythe, if you can share your perspective.
Yeah, I mean, I would follow up on both what Fred and Harry said. I think even though there are so many great opportunities, and we're thinking about it at Tune on how we could target epigenetic editors for longevity and for health span, you do actually have to pick a target or a set of targets that you can have a really clear endpoint on to be able to read out. So we're thinking about cardiometabolic, or we're thinking about diabetes. We have to have clear serum readouts. We have to be able to look in a biopsy and say, yes, we've actually changed the epigenetics here from pre and post, and it's durable, versus if you're looking at a whole set of downstream sequences and changes in epigenetics, it's going to be a lot murkier from your endpoints. So for us, because we're a new modality, we're kind of starting out there. That's one of the reasons we started with HBV, is because it was a very clear way to show the modality works. And then as we shift to cardiovascular, we think about known targets and pairing those up and multiplexing them and being able to look at those readouts. And then we can get to more complex diseases and thinking about longevity. But I think you have to, with these newer kind of approaches, you really have to pick something that you can really read out clearly.
And Ben, I will turn it over to you.
Yeah, and I think, the inflammation piece it's such a wild ride, especially from a Lilly perspective. I mean, we've spent a lot of time sort of reviewing and had our own programs, obviously looking at things like canakinumab and obviously the wonderful things it did. However, again, talking about infection risk and other sort of AEs and long-term risk obviously scared us away from some time. But I think now, given the fact that we've got wonderful drugs in the cardiometabolic system that can lower triglycerides, lower LDL, there's still this problem of, again, pushing that relative and absolute risk ratio down further. And we know that you can basically flatten out triglycerides, you can flatten out LDLs, but it's still not fixing the problem. And again, it's opening the door of, okay, it's probably inflammation and something else too, but it's allowed us to get back into this space. And I think it's intuitive, or at least at this point, it becomes imperative that we start doing it. So we've obviously made deals where we're looking back again at NRLP3, we're looking at IL-6 as methods. But one thing too that's been nice about this is now that technology is getting to the point of maybe we don't necessarily need to worry about sort of systemic fires, we can put out local fires, and again, ask that question of, if we were able to deliver specifically to liver, to the heart, to the skeletal muscle, or even bone itself, put those fires out, ask that question of, if we can reduce inflammation sort of compartmentally, does it also have a benefit? And again, now that there's probably tools, whether it's siRNA, genetic editing, genetic therapies, that could allow us to bring sort of even stronger weaponry to really ask that question and test these hypotheses. And I think now we're getting to that point, and we can do it in isolation. Yes, the hope is we do get it sort of more broadly, more systemic, but at least we can start asking that critical question of, does reducing heart inflammation, or does reducing even beta cell inflammation promote overall function and viability over time? And that's, I think, what we're really excited about. And again, that makes a very clear path forward as we start thinking of phase 1A and phase 1B trials, because as you know, a lot of these trials are very difficult, and sometimes it takes a lot of time for you to appreciate fibroinflammatory mechanisms and how they actually manifest into something meaningful that you can measure.
All right, well, that's a good segue. Maybe we can shift gears from the underlying biology to the actual clinical signs and symptoms that show up for the aging patient. And we spoke a lot about the cardiometabolic disease, and heart disease is still the leading cause of death. Neurodegeneration is something that we will all experience if we live, if we're fortunate enough to live long enough. Sarcopenia, frailty, fibrosis, declining immune system. Maybe I'll add cancer to that list, since the exciting results at ASCO last weekend. But there's so many of them, and I think as investors, a lot of them are underappreciated. So maybe we'll ask our panel to pick one that they think is especially underappreciated and a validated target that they think deserves a different clinical development strategy. Maybe we could start. We'll go this way this time, starting with Ben, and ask each of you to identify one.
Now, for those who know me, knows I'm super biased and passionate about this. But I think, for us, I mean, I'm going to say muscle just because obviously it's my background. That's what I was trained in. So again, I'm always going to give it a prop when I can. But I think, in terms of longevity, I think it's one of those that, the first time we went around in this space, 10 or 15 years ago, where we had things like myostatin, we had SARMs, we had some basic mechanisms that we knew built muscle. No question about it. They built muscle wonderfully. They got a little bit of a bad rep, obviously, once we got into function. Again, we know regulatory agencies were very stringent on the criteria that was going to be required for them to move forward. But it wasn't like these, I always say to people, it wasn't like the drugs completely failed. We did see evidences that, yes, aside from mass, there were improvements in certain components of the SPPB that weren't appreciated at the time. But again, because it didn't hit all of them, we weren't able to move forward. And then at that time, it was like, hey guys, you didn't do it, move on, muscle's dead. So I think it's one of those that, again, kind of going back to the earlier pieces that, understanding what having more muscle mass looks like. And again, I think there was a comment earlier about kind of going downhill and not forcing the body. So I think Yury and I were talking, it's like, I actually want to force the body. I want the body to work a little bit more because I think the act of building more muscle, the act of building even more bone, hopefully indirectly, is a great thing. I think it's one thing, again, with resistance training, we know this is that forcing anabolism and forcing the growth of that muscle allows for turnover, allows a cleanup, it allows the remodeling process to cure. And I think eventually over time, you would see those more appreciative quality aspects. And I think it's a wonderful piece. And not to mention, you burn a lot of energy. I mean, building muscle takes a lot of ATP. It's one of the most expensive processes in the body. So the longer you do it, even if it's not continued growth, but even maintaining growth, I think from an energy expenditure, from a body comp, just metabolic benefit, I think it's going to be overall beneficial. Again, how you start describing things like function, we're working with every great institute in the world and experts to find better ways to communicate to regulatory, obviously officials and whatnot, how to move that forward. But I still think, one, there's mechanisms there, even the ones that are underappreciated that didn't maybe show everything they want, that are still going to be very useful. So that's, again, my very biased opinion on it.
Excellent. Blythe?
Yeah, I'm also biased, obviously, in working in the epigenetic space, but I do think that thinking about tissue regeneration, particularly in the liver and in the pancreas, there's a lot known about what the epigenetic changes are for both fibrosis, in diabetes, in cirrhosis. I think actually understanding those epigenetic changes and being able to harness kind of some of these tools to be able to drive changes, to drive regeneration, I think could be a really, really powerful way to kind of take some of these new drugs, these new epigenetic and genetic drugs, and actually apply them. We know we can get them to the liver. So that's, with the muscle, one of the big challenges is getting to the muscle. But with the liver, you can get there, and both the liver and the pancreas, I think, are both places that would be really great ways to apply some of these drugs.
Great. Fred?
Yeah, I'm gonna kind of side with Ben on this. I think, as we age, one of the biggest issues is frailty. And, we know that the older you get, the harder it is for you to tolerate the current metabolic treatments for like the GLP-1s and whatnot. So something we can do to not just increase muscle mass, which is obviously a goal and an outcome that we want to measure, but, the functional ability of people to stay active. That's really, I think, one of the keys, and certainly I saw it with both my parents as they got older and before they passed. They really wasted away, and at the end of that time, it wasn't so much obesity that was an issue, it wasn't diabetes that was an issue, it was frailty, and you're one step away from falling and breaking your hip, which is often the end of life for many people. So I think that's one of the most important things that we can work on for sure. And right up there with it is neurodegeneration, because the hardest thing to do is see somebody lose their mental functioning when they're otherwise healthy. So couldn't really choose, but those are the things at the top of the list.
Yeah, I think aside from all the great points around frailty, if I was to answer it in two parts, I'll start with sort of a biological area, then something around clinical trial design. I think within biological area, we are very, we made an investment in the fine regeneration space, which until relatively recently I think was massively underappreciated, but that seems to have made a bit of a resurgence. And I think that also has quite a lot, broader immune reconstitution. I think, if you can create sort of an immune restabilizer in the way a GLP-1 is a metabolic restabilizer, I think that'll be pretty important for age-related disease. But I think separately to that, we've been thinking more around how do you demonstrate all the benefits of a longevity mechanism in a clinical trial that doesn't require you to have a very long outcome study, or whether we can get to a composite marker of biological age and by that. And we've been looking at whether you can design sort of secondary, tertiary endpoint study designs that can demonstrate this multi-organ effect of a longevity drug, which, in my opinion, if you have a drug that's acting on aging biology, it should probably be effective in more than one organ system. So I think that's something which should be interesting to see. Are those some pretty easy wins we can pick up for cheap in a clinical trial, just through a blood draw or a scan, and then we can kind of pull as much data as we can from that. And I think there should be some more attention spent towards that.
The downside of being at the end is all the good ideas are taken.
That's what I was thinking.
But I think, Paul, you will have the worst bit. But I think we should be quantitative here. If our objective actually is to improve longevity at a public health level, we've got to look at what is the number one killer. That happens to be cardiovascular disease worldwide. And in this country, it actually is getting worse. That's despite having more LDL lowering and hypertension lowering and everything else. So there is a very definite need to address what is increasingly called the residual inflammatory risk. I think Ben alluded to, and I think that need to be addressed smartly. When I say smartly, that means one has to demonstrate clinical efficacy on top of standard of care, like GLP-1, like SGLT2, and that's sort of data that I was presenting just last week. So we actually made patients lose weight with calorie restriction, and then our drug on top to see how much more inflammation can be reduced. So I think there's a lot of hand waving and vaporware around longevity. We have to break it back to good science, good peer-reviewed science, that actually we can stand on the fundamentals on.
Yeah, so I agree. I think one important aspect, and there is an opportunity, is if you look at conditions associated with aging, in many cases they're not even considered pathology. If you or your parents or someone that's older goes to a doctor and is anemic, and you happen to be anemic, the doctor says, "Well, you're just old. You get a low blood count." Or you're malnutrition. Well, you don't eat as much because you're just old. Or frailty. Well, you're a little weaker. You can't do what you've done previously. Things are not even acknowledged as pathology, and by correcting them, you could have a huge effect on morbidity for those people. So the first thing is education, I think, is an opportunity to realize that these are actual pathologies that can be treated and can improve outcomes. Another thing is, rather than choosing something a priori, one of the things that's being mandated, especially for heart disease, is that we're obligated now to do these outcomes trials. These trials are tens of thousands of people going for years, sometimes 3 or 4 years, costing billions. Yet why do we only focus on the cardiovascular outcomes? You've got a captured elderly audience that you're looking at longitudinally for multiple years. And why not attach other observations to this? You can look at the incidence of cancer, you look at the incidence of cognitive dysfunction, you could look at osteopenia, you could look at kidney disease, all these things, and determine if the mechanism that you're evaluating for cardiovascular disease also has a benefit for these other things. So make sure that those are written into the trials. They're in there. You can look at these data. These are the first time ever that we're doing these longitudinal trials where we have an ability to look at more than just the hard outcomes.
I think that's, Paul, also a very good point to talk a little bit about how we can, from a regulatory perspective, develop those drugs. And, aging is not recognized by the FDA as a disease indication, and at the same time, we don't have a very good, or at least well-established biomarkers. Or maybe we have, let's discuss, but in aging. So, and then if we look at the big age-related disorders, those clinical trials in those diseases are pretty long, very expensive, and it might be very difficult, for a relatively small company to run those trials. So I wonder what kind of playbook are you considering in that regard to basically de-risk those programs, to make them more capital efficient? And I don't know, like the things which are coming to my mind, like adaptive clinical trial designs, I don't know, maybe surrogate endpoints, maybe like collecting additional biomarkers, or maybe just, stratifying patients in some of the disease indications we know really well. But first, maybe, Paul, you can comment on that.
Yeah, so I think the key for regulatory approval, obviously, is to develop some type of manageable biomarker or surrogate endpoint, I think, in looking at it. We can't do, in tens of thousands of patients, we can't do coronary artery catheterization on all 10,000. So, although maybe we can. But I think that understanding biomarkers, especially, and there are examples, for example, if you look at the treatment of hypercholesterolemia, that has become an acceptable practice while looking at the surrogate as opposed to actually looking at the outcome. So can this then be applied to aging-related diseases, because there are no good mechanisms. So I think a good example here is for cardiovascular disease, especially related to inflammation. We know that CRP is elevated. It's a clear surrogate that appears to correlate with outcomes, but it's not an acceptable regulatory endpoint at this point in time. So how do we get these types of markers to be accepted and ultimately be approvable endpoints? Right now, it's just vague. It's sort of, suggest a bunch of things to me, and I'll let you know if one strikes my fancy. And that, for right now, that's unfortunately becomes very difficult.
Mm-hmm. Yeah, thank you. And Jyothis, during the prep, we discussed, that there is, according to you, if I'm citing you correctly, that there are three things the FDA really cares about. And actually, I forgot already what were those. So if you can, answer the question and maybe also highlight those.
I know there is a vacancy at the FDA, but I'm not applying for that, and they haven't asked me either, and therefore I can't officially speak to it. But it's a well-established FDA principle, as well as EMA and MHRA in the UK. They look at three things: feel, function, and survive. So I think Paul was talking about the CV outcome trials for survival. How many dead bodies do you count at the end of the day? And then you tally up. That's the survival ultimate benefit, and that applies across all divisions across FDA. The other thing actually is feeling, like there is increasing emphasis under the current administration on having validated patient-reported outcomes that actually can contribute to approval. And the third thing is actually function. I think, it's good to have a big muscle or a big bone, but what does that lead to in terms of function? And here, we all criticize the FDA, but they've been actually very good at heart failure, approving drugs based on 6-minute walk test and KCCQ and things like that. There is actually a lot of momentum there. So I think, therefore, we can all wait and protest and the world has to change until we do anything. Or we can actually, just like SGLT2, GLP-1 and the LDL world, you can actually plan for a land and expand strategy, as I call it. That is, land with some functional outcome or a patient-reported outcome benefit. Then you expand into the longer-term, bigger studies to show hard outcomes. That's been the playbook over and over again in diabetes. For example, we got drugs approved on A1C lowering. The money was coming in at some point, and then you go and do the CV outcome trial. That's how EMPEREG and the early GLP-1 studies happen. So and I know you are a big fan of biomarkers, but I think, and I love a CRP to be an approval endpoint, but the difference between a CRP and HbA1c, for example, is there are two well-controlled trials, DCCT and UKPDS, which actually have shown that lowering A1C leads to better outcomes. And same for LDL. one day we will actually be in a place where CRP lowering improves hard outcomes. We are not there yet. So until then, we have to play by the rules of today, I think.
Yeah, no, I think it's an excellent point about A1C. That was such a milestone, which was super important to develop so many drugs, and hopefully we will get more of those for like surrogate endpoints for the healthy aging. I will turn it over to Blythe if you have anything else to mention.
Yeah, I mean, the way we think about it at Tune is, if you're thinking about in CV, really harnessing known biomarkers that the FDA accepts. So looking at LDL and things like that, we're targeting some of these genes where we're repressing them really with a transient drug. So we actually have the benefit of you give the drug one time, you're looking for changes in these biomarkers that are durable. And if you have that durability for 12 months, it actually signals to you very quickly and efficiently that you could do the longer term studies to actually look for more of these secondary endpoints and these survival benefits. So in new modalities like this, where it's really you're not hung up on having to make sure that the patients are compliant on the drugs and that sort of thing, it actually gives a benefit of having a better, more efficient trial.
Excellent, thank you. I will turn it to Jay.
Yeah, thank you. Really appreciate the pearls of wisdom from our panel here. For those aspiring entrepreneurs in the audience, we thought it might be interesting to seek our panel's advice in terms of, and also aspiring investors, not just entrepreneurs. So both sides of the equation. Any advice that our panel would like to share with us in terms of thinking about health span for investments or for entrepreneurship, raising money for health span biotech ventures. I guess what advice would you give to either investors or entrepreneurs raising money? Maybe Ben, we'll start with you and work our way down.
Yeah, I mean, for us, I think as we get into this space, and luckily I've been annoying enough over the last few years to push Lilly into some of these areas, and again had some great interactions, obviously, with companies like BioAge and whatnot. We were able to sort of truly understand what these relationships, can really become and how they can produce sort of, step forwards into the field. And I think specifically for us though, aging's always tough. So anytime we bring aging in, it's going to be, what is the question that we're asking, right? And so as we convince our key stakeholders and our senior leadership, everybody loves aging, no one's challenging, but what's my phase 1 look like, right? What are the biomarkers that are going to be able to help us ascertain is at least doing something that it's supposed to? And what's the work plan to get you there? And I think what's great about that is, again, we try, hopefully Paul will corroborate this, but I think, we try to work as closely together to understand what are those next steps? What is the plan? Again, we want to make it effective too. We don't want to, because you could do 95,000 things in preclinical animals and IND enablement to get you there. But again, the goal is to get it to the clinic as fast as possible. And then we can start really identifying and sort of querying what's happening at that point. So it's really that work plan. It's really that work. If it's an asset, it's a work plan on how to test it and what we're going to test in phase 1B and eventually what a phase 1/3 or phase 3 looks like. But if it's databases, all this, it's also, why is yours unique? How is it going to help us ascribe more value to a particular target? How is it going to be able to differentiate from what's out there? How is it going to complement what's already out there? So being very, at least specific on that sort of hypothesis is key, because you got to remember, when we pitch things, we're pitching again, we're Lilly, so we're going to see 1,000 different things. So that one thing is going to have to step out, especially in aging. And I think having a clear, concise work plan is always the key. And again, the great thing about what we do is if there's success, we're going to maximize the value as much as possible. So, but get us that little hint of activity that it's doing something in a clinic, and then we'll set a nuclear reactor to it at that point. So,
All right, super helpful. Thank you, Blythe.
Yeah, I would echo that, like having a really clear plan and readout. So if you're looking at something, say like liver regeneration, what is your quick primary endpoint? How are you going to show you've improved function? In the case of what we're doing for epigenetic reprogramming, how do you show you've actually done what you said you've done and not some other thing, some side effect in a short amount of time? I think one of the other challenges we have as a new modality is being able to model these systems in animals. Particularly epigenetics are not, necessarily one-to-one when you go from a mouse to a monkey to a human. So when you're thinking about a new modality, really thinking hard about what your model systems are there so you can prove things out and get to the clinic quickly.
Great, thank you. Fred?
Yeah, I'll kind of combine it with the last question too. So I spent the first half of my career at a company called Allergan, working on a product called Botox, and I was eventually the chief medical officer there. We got approvals for that drug across the globe in numerous conditions. Of course, everyone knows about the cosmetic use, right? We actually made more money on the therapeutic use, but people don't realize that because we had, I don't know how many indications we had on the therapeutic use. And I see the longevity space, some similarities there. Because imagine, going to the FDA to say, we want to get a cosmetic indication for the most potent neurotoxin known to man. These are tricky conversations, and the regulators, they're not there with longevity yet. And frankly, half or more of the people talking about longevity are charlatans and quacks, especially on social media. So you've got to have your feet on the ground, land and expand. You go with an indication. I think investors want to hear about an indication that the FDA accepts, where there's a regulatory pathway, where there's a market you can assess. And yeah, you can have a longevity drug in the long run, hopefully, but start with something that the FDA is going to accept, and then build your case from there. And that's what we're doing. And so I joke and I say, I spent the first half of my career making people look younger, and I'm going to actually make them younger. But I'm going to get there through a disease that the FDA recognizes.
Excellent. All right, thank you, Fred. Harry?
Yeah, I'd say similarly to being very focused in going down a certain indication route to get that sort of clinical tractability, but also that pairs with, a good business development case and ultimately liquidity for your investors, which we'd like to think has a bit of a flywheel effect on the entire field. But I think, just to add maybe a different perspective is, I wouldn't skimp out on doing the fundamental molecular biology work early on, particularly around determining the mechanism of action of your drug, because, that could probably still hinder you even if you get further down the line. And I always appreciate it when you come across a company that's done the hard yards up front early and really understands how their drug works and can articulate how that links to the biology of aging. So I'd say that's probably the biggest thing that I would take away is do that work early.
All right, thank you, Harry. Jyothis?
Yeah, first of all, I want to agree with Fred and Harry. There's a lot of, you call them charlatans, you call it poor science, but I think you often find, lots of press releases on the same 3 patients or whatever, right? So I think that is one thing to really be very careful and not to do, in my view at least. But I think I have this 5P principle of what a good pitch would look like for this sort of thing. The first P is patients. How would you actually add value to those patients? Can you explain to your mom and dad if that's your patient? The second is a prescriber. Your prescriber's life is already pretty bad. And you're asking them to prescribe this new drug. What value is that person going to get? The third thing is the provider system, P number 3. I mean, healthcare systems are running out of money and are barely above water. So you're asking them to adopt to innovation. What isn't it there for them to adopt to innovation? The fourth P is policy and public health markers. We actually violently say we should recognize obesity as a disease, aging as a disease, but what is the actual public health payoff? We rarely articulate. But the biggest P is the payers. Who's going to pay for it? Is it a public payer? Is it a self-pay? And if so, what is the value proposition for that person? If you don't have those 5 Ps lined out, I think you can actually get a great regulatory approval, but that will be a Pyrrhic victory, the 6th P.
All right. Thank you, Jyothis. Paul?
Yeah, I think, again, I'm suffering from the end of the table syndrome as well. There's a couple things. One is, if you're investing in it, how de-risked is the program that you're actually going to be investing in? Is there human data that's correlative? Are there animal models where these animal models have previously been validated in terms of human disease? Is there genetic evidence in humans that this is actually something that is viable and doable? There are very few mechanisms that you can go directly from a mouse to a human and say that this is going to work. I mean, everything works in a mouse in looking at it. So in trying to get this to be
de-risked as far as a human perspective, I think you really need to look hard at the data associated with this. And now, fortunately, we have ways to get human first data. A good example is said, we can do proteomic analysis on patients, pre-morbidly, and then that will predict what actually happens. So is there human data, I think, is the most important aspect of it. Secondly, and I think it's been touched on, is what you're intending to do practical. Do the patients even exist? Good example, we were interested in looking at anemia associated with aging, and we tried to do a study. We couldn't get a single patient enrolled in a 6-month period of time, and it seemed like it was a common enough disease. So are these studies actually doable is another aspect that you really have to look at, and just practical in general. So it's not the science necessarily. Everyone has great scientific ideas, but it's a question of whether or not what you're intending to do is actually possible to do, and make sure that you believe that before you make an investment in something.
Excellent. Thank you so much, Paul. Thanks to all our panelists. And Yury, it's been a pleasure. I'll turn it back over to you for closing remarks.
Thank you, Jay. Actually, the time is up, but we almost perfectly at the time. And I would like to thank all the panelists and again, Sachs Associates for inviting all of us. That was a great opportunity. And thank you, everyone. If anyone has questions, I think we're all available to answer after the panel.
Well done.
Thank you so much.
Thank you.